Hello, this is Sahana here with the topic dominant epistasis. So, dominant epistasis can be simply defined as the prevention of the expression of a gene by a dominant non-allelic gene, or when the dominant allele of one gene masks the effect of either allele of the second gene, it is termed as dominant epistasis. So, in this particular situation, the classical F2 phenotypic ratio 9 is to 3 is to 1 gets modified into 12 is to 3 is to 1 ratio. So, this is the most important. So, the dominant epistasis phenotypic ratio, that is F2 phenotypic ratio, will always be 12 is to 3 is to 1. So what actually happens here is when dominant allele at one locus, for example, say A. So this A allele produces a certain phenotype regardless of the allelic condition at the other locus. So this A is called as, in this particular situation, A is called as, or A locus is said to be epistatic to the B locus. And further, since the dominant allele A is able to express itself in the presence of B or this B, this is a case of dominant epistasis. Alright, so only when the genotypes of this A, this individual, is in homozygous recessive condition, as you can see here, only then the genotypes capital B or B, this can be produced. So thus what can be told is, to consider these genotypes, the capital A and capital B, when there is at least one of the functional enzyme A, capital A, is produced. If these are produced, then the phenotypes will be same. That is, the phenotype will be of the allele A. If the functional allele is absent, or the functional allele, that is A, is absent, then the phenotypes that is produced are depending on capital B or small b. There two different types of phenotypes are produced. So what is happening here, when these two genotypes are present, the phenotype will be same. That is, one phenotype is there and two different phenotypes are being produced here. So totally three phenotypes. That is how we get 12 is to 3 is to 1 genotypes here. So to consider an example, this is a one parent which produces purple color product, and this is another parent which produces white color product. And what happens in the F1 generation, there is a production of F1 product again, okay, which is same as one of the parent. And when this is being self-crossed, the F2 self-crossed progeny has 12 is to 3 is to 1 ratio of purple, red and white respectively. So in this particular example, what can be understood is two non-allelic genes are interacting, because the F2 phenotypic class so obtained is less than 4. As you can see here, 12 is to 3 is to 1, only 3 phenotypes are being produced here, which is lesser than 4, which is lesser than the classical 9 is to 3 is to 3 is to 1 ratio here. So this particular phenotypic ratio can be explained using this particular pathway here. There is a white substance which is called as substrate. So in this case, enzyme A and enzyme B compete for the same substrate, that is white substance here. So here enzyme A, which converts the substrate white substance into purple product, has much higher affinity to the substrate than the enzyme B. Okay, so enzyme B, this converts the substrate into red colored product, and the difference between the difference in the affinity between these two enzymes uh, is so marked that enzyme B can only work efficiently if enzyme A is absent. Otherwise, if they are both are present, only enzyme A expresses itself. Only when the enzyme A is absent, enzyme B can express itself. That is, allelic condition also depends here. If the dominant allele is present, enzyme B will produce its color. If the recessive B is formed, then the product remains the same. Or if any one of the allele is dominant A, irrespective of this particular allelic condition, then the substance will always be purple product here. So this particular slide explains you the genotypes of the F2 progenies. So here I told you 12 is to 3 is to 3, 12 is to 3 is to 1 ratio, right? So here 12 of them are purple here. So all these genotypes have at least one functional allele A. As you can see here, all of these have at least one at least one functional A allele that converts all the substrate into purple product. Whereas in this particular scenario, what's happening here is there is no functional enzyme A. In both of these cases, there is no functional enzyme A. So what happens? So B can express themselves now. So here it is homozygous dominant B which can produce red, and it is heterozygous B which can also produce red color here. And here it is white in color. Why? Because there is no functional group A or there is no functional group B. So both of these functional enzymes are absent and which cannot synthesize any colored pigment here. So dominant epistasis being discussed here. There are few examples of or few genetic problems to make us understand the concepts better. So here in sorghum caudatum, which is commonly called as cholam, the inheritance of grain color is being studied here to understand the or to represent the dominant epistasis. So what actually happens here is there is the pearl grain color which is dominant over chalky. So there is a grain color called pearl grain color and chalky grain color. So what happens, what is the relationship between these two? Pearl grain color is dominant over the chalky grain color. And when will this pearl grain color develops? When the dominant gene Z is present. And the chalky color develops in the absence of the gene Z. So the relationship between these two are just the allelic interaction is being happening here. When the dominant Z is present, the color is pearly, and if it is recessive Z, then the chalky color is being produced here. And we'll see what happens next. Another dominant gene W, which is responsible for the red color of the seed, is dominant over gene Z. So whatever the gene, dominant gene W is coming into picture here, this is the non-allelic gene that we discussed about. This is the non-allelic gene that is present in the different loci or the other loci and has the effect on the allelic genes here. So this is responsible for producing red color of the seed, and this is dominant over Z as well. Andre, it is dominant over the dominant Z as well as the recessive Z here. And when W is present, that is the dominant gene or dominant allele W is present, the grain will always be red in color irrespective of the presence of the other gene Z. So I told right while explaining the dominant epistasis. So the capital W or the dominant allele W will always produce its trait irrespective of the presence or absence of the other non-allelic genes here. So in the absence of dominant allele W, the green color will be pearly or chalky depending on the presence or absence of the gene Z. So this is a introduction of this particular inheritance, and the question is like this. When a red colored seed type is crossed with a pearl variety, the F1 offsprings are red in color. They have given the F1 offsprings, and then they are telling us to give, they're also telling that F2 generation shows all the three kinds of seeds here, that is pearly, chalky, red. All the three varieties are being produced in the F2 generation. So this is the question here. So we'll have to put up the cross for this particular question here. So before moving on to the solution, or just starting up with the problem, we'll have to analyze what is being given here and how to take the genotypes and how to make up the crosses and all. So for that, we'll have to analyze here. So as I said here, just read sentence by sentence. Okay. So what does the first sentence say here? The pearl grain color is dominant over chalky. Okay. So pearl grain color is dominant over chalky. So that is being understood here. And the pearl grain color develops when dominant Z is present. So what can I take here? I can take the dominant Z produces pearl grain color, and chalky color develops in the absence of it, in the absence of dominant gene. So, in the absence of dominant gene, which means the recessive allele Z will give you the chalky grain color. And what does the next sentence say? Another dominant gene W is responsible for the red color production of the seeds. So the dominant W will produce red grain color. That is being analyzed here now. And what is being said here is, so here W is present, the grain will always be red, which means W is dominant over the dominant gene Z. So I have written here W is red color. This is a non-allelic gene. This is dominant over both of these varieties, that is pearly and chalky. So this is dominant over both of these alleles, and Z is dominant over only the recessive allele Z here, which means the dominant variety, red is the dominant variety when compared to these two, and pearl is dominant to chalky while it is recessive to red color. So now we'll jump into the process here. So the F1 parents are like this. The phenotypes are already given in the question, that is red and pearl. These two varieties were crossed. Now while taking the genotypes, it plays a most important role here. So pearl. If I need a pearl variety, then I'm sure that there should no functional W allele here. Okay. So because the question says the W, the dominant W is present, the grain will always be red irrespective of the presence of the other gene Z. So keeping that in mind, I should always take small w or recessive homozygous W with the dominant Z. Only then the variety will be pearl grain variety. And while coming to red obviously it is like any one of the W allele should be dominant. So I can take both of them dominant and these two recessive because this is already there here. And F1 gamete formation, one of them goes to one gamete and it goes like that. Gamete formation happens, and when they combine, F1 generation hybrids are produced with double heterozygous grain color genotype here. So here both of them are present, that is dominant W is also present, dominant Z is also present. But with the depending on the analysis, whatever the analysis we did, W is more dominant than the pearl. So the hybrid produces the grain color red here. Okay, irrespective of the presence of this particular dominant gene Z, the F1 hybrid produces red colored grain. Now to further analyze the crosses, or to further carry out the crosses, I'll self cross these two F1 hybrids. So that becomes the F2 parents here, and then gametes are formed, and then I'll be taking the gametes on either sides of the Punnett square here. And when the combinations are made here, and I'll fill the genotypes first. Okay, to avoid confusions, I'll fill all the squares with the genotypes first. I'll combine these two. So these two, if I consider capital W, capital Z, and these two, capital W, capital Z, small z, and so on. Every squares will be filled with genotypes first, and then to estimate or to predict the phenotypes, I can take help of this particular analysis here. So red is whenever a functional allele W is present, the substrate is converted into the dominant color here, right? So a dominant substance here. So the dominant color is red. So here W is present red. Wherever the at least one of the dominant allele W is present, then I can take it as red. So all these four are red. These are red. This is red. This is red. This is also red. This is also red. While when these functional enzymes are not present, W is not present, dominant W is not present, then only then I should consider the Z, the allele condition of Z. So here it is homozygous dominant, so the dominant variety is being produced, that is pearl variety, and then this is also one of the dominant Z is present, so this is also pearl variety. Here it is the same pearl variety, whereas here both the functional enzymes are absent, or both the dominant alleles are absent here. Both of them are homozygous recessive, so there is no color production of the grain, so it remains white. So that is how we get 12 of them red and 3 of them pearl and 1 of them with white color grains. So this is the combination or the phenotypic nature of the F2 generation hybrids. So this is most important thing, the phenotypic ratio of dominant epistasis. So this is like 12:3:1 is the ultimate F2 phenotypic ratio here. And this is the second example. So in squashes, summer squashes, which is cucurbita pepo, there are the inheritance of fruit colors is considered here. So there are common fruit colors which are of three types. Okay, the white color, yellow color, and green color. Of these three fruit colors, the white color is dominant over both yellow and green. Okay, so the white color is the dominant over yellow and green, and then yellow is dominant over green only, and yellow color fruit is dominant over green alone. So the question is, in a cross between white variety and a green variety, the F1 progenies appeared to be white fruit type. Okay, so the white variety and green variety is being crossed with the F1 hybrids as white fruit type, which on selfing gave 12:3:1 ratio in F2 generation, and the type of interaction was of dominant epistatic type. So these are the different types of question formations. So here it's the same. Now we'll analyze the question here. So what is being given here is there are three common fruit colors, and white, yellow, and green are there. I can take dominant allele W. This will be the non-allelic type. So capital W will give white fruit color, and I'll take capital G for yellow fruit color and small g for green fruit color. So this is clear now. White is dominant over green and yellow is dominant over green, whereas it is recessive with the white colored fruit. Now this is the cross. The question says the cross was made between white and green parents, or white and green colored fruits, right? So the phenotype is being written here, and genotype, again we'll have to be careful. Here green, when will the green color be produced? When all the alleles are recessive here. So I'll take all the recessive genes here. And when will this white be produced? When the functional enzyme W is in dominant condition. So I'll be taking all this homozygous, both of this as homozygous dominant. And the gamete forms F1 generation gives white colored fruit. So though this is double heterozygous in nature, I should always take white fruit color, because why? This particular uh, enzyme, or this particular allele that produces enzyme W has more affinity to the substrate, right? So this gives white color to the fruit now. Now, the F1 parents are the F1 hybrids when they are self-crossed. So these are the F2 parents here, and the gamete formation happens. I'll take them in the Punnett square. I'll find out, or I predict the possible outcome of the progenies here. So here, the same as the first genetic problem, whichever has at least one of the capital W or dominant W gene, they produce white fruit color. Alright, and whenever these dominant gene W is absent, only then I can consider the G alleles. Okay, if the G alleles are homozygous dominant, then their color will be produced, that is yellow. Here one of them is G, dominant G, so yellow color is being produced. Here is also one of them, one of the allele is capital G or dominant G, then the dominant trait yellow color is being produced. Whereas here both the functional alleles are absent, that is both the dominant alleles are absent, all of them are recessive homozygous, so green color, that is the recessive trait, is being produced here, and ultimately living with a 12:3:1 phenotypic ratio with the phenotypes white, yellow and green. So that's about the dominant epistasis. I'll be back with recessive epistasis, which is a type of epistasis in the next class.